Hello and welcome to this revision video on the whole topic of biodiversity for OCR A-level biology. So we can define biodiversity as the variety and complexity of life. And this could be life in one particular ecosystem, or it could be life on Earth, so the whole living world of the planet. There aren't really any strict boundaries. Now, we can consider biodiversity at three different levels. At the level of the genes, at the level of the species, and at the level of the habitat. So when we're thinking about genetic biodiversity, we're referring to the variety of genes and their alleles in a gene pool. A gene pool is the collection of all the genes and their alleles in a population. And since we're talking about populations, genetic biodiversity is always displayed between individuals of the same species, because by definition, a population is a group of individuals of the same species in a particular habitat at a given point in time. If you remember, in a previous topic, we looked at intraspecific variation and how it has a genetic component to it. Well, we can refer to that as genetic biodiversity as well. Now, here's one key idea. A population with high genetic biodiversity will be less susceptible to extinction than a population with low genetic biodiversity, and we'll find out why that's the case in just a few moments. We can calculate a value for genetic biodiversity, and we use this formula. The number of polymorphic gene loci divided by the total number of loci multiplied by 100 to get a percentage. So what are polymorphic gene loci? Well, the locus of a gene refers to its exact position on a chromosome, its specific location on a molecule of DNA. And if a gene is polymorphic, then it has many alleles. That means that on its locus you can find many different sequences of bases. Many different alleles can slot into that one position, that one gene locus. The total number of loci, of course, refers to the total number of genes, therefore. And then this formula, this value that we obtain, gives us an idea of the genetic biodiversity of a population. If the proportion of polymorphic gene loci is large, that means that there are many genes with many alleles, and that means that the gene pool will be larger, and therefore we can say that the population has high genetic biodiversity. Species biodiversity has two components. This is the range of species and the number of individuals of each species in an area. The first component is called species richness, and this is the number of species in a habitat, the count of species. Whilst species evenness refers to the relative proportion of individuals of each species in a habitat. So let's say that you're looking at a particular field somewhere maybe, and there are 15 different species present. The number 15 is your species richness. If let's say that there is a lot of individuals of one particular species but very few of everyone else, then you're going to have low species evenness, because there is a skew, an uneven distribution, or rather an uneven proportion. There is a lot of one species but very little of other ones, so it is not even, and so overall there could be low species biodiversity. Both of these components are important. Likewise, we can calculate a value for species biodiversity, and we use Simpson's index, which is D equals 1 minus sigma, which is the sum of, and then we have lowercase n divided by uppercase N all squared. So uppercase N is the total number of individuals in the community. It would be better actually to use the term community here. That is a group of all the interacting populations in an ecosystem. So that is basically the total number of individuals that you are studying. And then lowercase n is the number of individuals of a particular species. So with this quotient here, you are calculating the proportion of individuals of a particular species out of everyone else. You then add all of these fractions together and you subtract them from 1. And the answer that you will get will be on a scale between 0 and 1, where 0 is low biodiversity and 1 is very high biodiversity. And lastly, habitat biodiversity is simply the number or range of different habitats in an area. If you're looking at a whole country, for example, there of course will be very high habitat biodiversity. There are both forests and lakes and rivers and mountains and fields, many different habitats. Whilst if you're looking at a 1 by 1 square meter area of land, you'll probably only have one habitat, and therefore the habitat biodiversity will be low. And generally, the higher the habitat biodiversity, the higher the species biodiversity. This is quite obvious. One particular habitat might be able to be a home to many different species, and so the more habitats you have, the more species can access this particular area, and so species biodiversity will be higher. So now let's have a look at genetic biodiversity in a bit more detail. There are quite a few new concepts and key terms here, but actually you will look at many of these ideas in later topics in the course. So this is just an introduction for now. So I remind you, this is the formula for calculating genetic biodiversity. And so the more polymorphic genes that there are, the more alleles there are, and so the larger the gene pool of the population, which means that the population is more genetically biodiverse. So let's return to this key idea that high genetic biodiversity is associated with a lower risk of a species becoming extinct. Think about a major change to a population's environment. Some new, very harsh selection pressure, some new disease maybe. This is the classic example. If you've got a population with high genetic biodiversity, so a large gene pool, you are likely to find an allele which confers a selective advantage, which is advantageous in with respect to an individual being able to cope better with this major change in the environment. Whilst if you have a population with low genetic biodiversity, you're going to have fewer alleles in the gene pool, and you are less likely to find an advantageous allele therefore. And so if this major change to the environment does occur, then a population which does not have the advantageous allele will be completely wiped out. There will be no individuals of the population remaining, and therefore the species becomes extinct. Whilst if you happen to find this one allele, if this allele exists in even one individual, or maybe a few individuals, then those particular organisms can continue the population. They can carry it on to subsequent generations, and so the population does not become extinct. Yes, okay, everybody else dies, but these few people, these animals, plants, whatever, they can survive and reproduce. So there are a few factors that can affect genetic biodiversity, and again you'll look at these in much more detail in later topics in A2 content. You might remember from GCSE that selective breeding is the idea that farmers maybe look at their cattle and they select individuals with desirable traits. This could be high yield of milk or meat, and they breed those individuals together. They repeat this breeding process over many generations, so it is an iterative process, and eventually you will have a generation, a population with a high allele frequency for this phenotype, which is desirable. And this means that you have reduced genetic biodiversity if everybody has similar genomes. All the other individuals that had different alleles will have died. Captive breeding is something that we will look at later in this video. It is a method of conserving an endangered species in a zoo, for example. And then we have genetic bottlenecks, the genetic drift, and the founder effect. So let me illustrate these using a diagram. So here is a population's gene pool where each coloured circle represents an allele. Here is the bottle and here is the bottleneck. The bottleneck represents this catastrophic event, this major change to a population's environment. And you can see that only one allele will be able to pass through the bottleneck. Only this blue allele will be able to fit through, and only it will be able to make it through to the following generations. All the other alleles will be lost in the form of individuals dying as a result of this major change to the environment. And so this blue allele must have conferred some kind of advantage, allowing individuals that carry this allele to survive, reproduce and continue with the population. The founder effect is when you have a small group of individuals from an original parent population that migrate and isolate themselves, geographically usually, and establish a new population elsewhere. You can see that this new population has a lower restricted amount of genetic material to start off with. And so as this population continues to reproduce, to expand, the population will have lower genetic biodiversity than the original one because it had less genetic material to start off with. The only way that a new allele can be introduced into this population is either via gene flow or via mutations. And genetic drift refers to the random change in allele frequencies in a population, and this is all due to the random nature of sexual reproduction. Okay, now let's have a look at species and habitat biodiversity in more detail, and specifically at the features of ecosystems with high species and habitat biodiversity. Clearly, they will have high species richness and evenness, but also, interestingly, these ecosystems will have a moderate, hospitable and non-extreme environment, where the species present will have very few specific particular adaptations. This is because there is nothing extreme in this environment, and so there is no need to have any particular adaptations. And it follows from this that biodiverse ecosystems will have complex food webs and complicated trophic structures. And lastly, as we just saw, such ecosystems will be rarely affected by major changes to the environment. Even if one species is wiped out by a selection pressure, then all the other species can still survive because of the stability of the ecosystem. If you remove this one brick, this one species, then everybody else can still withstand this change because of all the cement, because of the large number of bricks that form this wall. Whilst if we're looking at an ecosystem with low biodiversity, then this wall will have fewer bricks and less cement. And so if you remove one brick from the wall, then it collapses completely. That's the analogy that we can use there. So here we have a specific example. This is the Sahara Desert, and you can see that we have camels there in the background. And we know from a young age that camels have very particular adaptations. The hump, for example, contains fatty material which can be respired to release energy and water. And then we have xerophytes as well, and as you know from previous topics, these are plants that are specifically adapted to low water availability. And so here the food webs will be very simple. The environment is of course quite extreme and non-hospitable. The tundra is another example of such an ecosystem. Polar regions are as well. But then compare this to maybe a tropical rainforest, which is of course very biodiverse, a very hospitable environment with a very complex trophic structure. Okay, now let's have a look at how ecologists can study the biodiversity of a particular ecosystem. So this involves a few practical techniques. Now it is fairly obvious that it is impractical or even impossible sometimes to carry out a survey of an entire ecosystem. That would mean that you'd have to count every single individual present. So that's every single plant, animal, fungus, bacterium and protoctist. That is never going to happen, of course. Instead, you would have to take a sample. and then you study the sample, you can do some statistics, and then draw conclusions about the whole ecosystem. For that, you have to hope that your sample is representative of the population. Now you might ask, well, what's the point of counting bacteria or fungi? Well, these actually play a very important role in ecosystems. They act as decomposers and they recycle nutrients. You'll learn about this in more detail again in later topics. So yes, you really need to know about every single individual present to get a good idea of the biodiversity of an ecosystem. But as I say, that is not possible. Instead, we take samples. So what type of samples are there? Well, sampling techniques can be either random or non-random. Random sampling is when each member of the population has an equal chance of being selected. You, as the researcher, have no involvement. You do not decide who will be in your sample. That is all done randomly and arbitrarily. For example, you can assign a number to each member of the population, and then you select your sampling units, the things or organisms that you're going to include in your sample, using a random number generator, or you can pick numbers out of a hat, for example. Now, of course, you can't number every single individual in an ecosystem, so instead ecologists might form a coordinate grid, and then they will randomly choose coordinates, and they will then place something like a quadrat at that coordinate and take a sample there. We'll have a look at how quadrats work in just a moment. Non-random sampling comes in three categories. The first type is opportunistic, and this is when you sample what is available at the time for you as the researcher, what is convenient for you. So you directly choose by yourself what you're going to include in the sample, and clearly this can introduce bias. You can yourself decide what you're going to sample purposely so that it supports your hypothesis. That is of course very biased, and it still unfortunately happens in science even nowadays. Anyway, the other type is stratified, and this is when your population naturally falls into categories, groups that we call strata. This could be age or gender or size maybe, and so you have to do a bit of calculation here. You use a bit of ratio and proportion to work out how many individuals you sample from each category, from each strata. And so the ratio of individuals of each category in your sample will be the same as it will reflect the makeup of your population. And lastly, you have systematic sampling, and this is when you sample individuals at regular intervals, either in time or in space. And the typical example here is a factory where you have a production line and you want to sample products at given intervals. Maybe you take every 20th product from the conveyor belt, or you come along every 20 minutes and then you take a sample. So it's this idea of regular intervals. Now, each of these sampling techniques has its own advantages and disadvantages, of course. The main problem is, of course, bias, and you need to try to avoid that as much as possible. To do that, you can make your sample as large as possible as well. With systematic sampling, one problem might be that, let's say, if you're taking every 10th product from the conveyor belt, then I don't know, if the machine in the factory causes a fault in the product every 7th product, then of course you will miss out on that pattern. Okay, so now let's have a look at what kind of techniques and apparatus we can use when we go out to do ecology fieldwork. First of all, let's have a look at how we sample plants. So we mentioned quadrats, and these are used to sample a habitat that you have already split into a coordinate grid. At GCSE, you kind of just said, well, you can randomly throw the quadrat. That, after all, isn't random. So instead, you have to use this coordinate grid. Now, you have two types of quadrats. You have these point quadrats. This is essentially just this rigid pole of wood through which you can put these metal rods. And then wherever the rod, the metal stick lands, onto whatever it lands is what you sample. And then you have these frame quadrats. You probably know how to use these very well already. You put them in a specific coordinate and you can either count the plants present and use percentage cover. You can use density, but very often it is quite simply hard to calculate every single blade of grass. So instead you just use percentage cover. You look at one particular box and you see if a particular species is present there. If it is, then you count that box. If it isn't, then you don't, and then you find a percentage. You can also use transects, and you tend to use this with systematic sampling, and you can look at how biodiversity, how species richness and evenness changes with distance. So here is a image of a transect. You can see you have this measuring tape, this line that stretches probably from the sea to somewhere else inland. And you can look at how a named biotic or abiotic factor changes with distance away from the sea, and therefore how biodiversity changes with distance from sea. You can use either a belt transect, and this is when you again use systematic sampling. You put your quadrat down at given intervals along the transect, or the other technique is called a line transect, and you simply look at what touches your measuring tape, what touches your piece of string. Now let's have a look at how we can sample animals. So first of all, we have a pooter. Basically, this is similar to a straw. So you blow into the straw on one side, and that pulls in insects from maybe a tree or from a bush into this pot at the bottom, into which all the insects collect. And there is of course a filter in the straw so that you don't swallow any insects accidentally. You can then use sweep nets. Classically, you just use these to catch butterflies, but you can use these on grassland as well. And then you can also use pitfall traps. This is when you dig a small hole in the ground, insert some kind of container, and then you need to add a little roof at the top as well, so that any precipitation or any leaves don't fall into your trap. And then any insects that are crawling along the ground will fall into the container. And then you might return in a few hours or in a day's time, and then you can look at what insects you have. Of course, all of these sampling techniques have many disadvantages, but this is as far as it goes with A-level, really. If you're going to sample aquatic animals, so fish or other invertebrates present in rivers or streams, then you might use kick sampling. This is very straightforward. You enter the stream, and in a particular place, you kick the bottom of the stream, the bed, to dislodge any insects, and then you collect them in either a sweep net or some other container. You can of course also use seining. This is when you have a net. Generally seining is used with fishing, where you have a very, very large net being submerged into the water. But again, you can use it to catch insects present in rivers as well. And lastly, if you want to sample or estimate sizes of populations of larger animals, then you use something called the Peterson capture-release-recapture method. So the process is as follows. This is a very interesting technique. You take a random sample of a given number of individuals in a habitat. Ideally, this is as large of a sample as possible. So you capture, this is the first step, you capture as many individuals as possible, and then you mark them somehow. You might put a tag on them, you might put a ring around them. If it's like a bird, you might put a ring around the bird's foot, or any way of just marking the captured individuals. You then release them back into the environment, and then you wait for some time for the individuals to, evenly spread out, to redistribute themselves in the population. And then you come back and you take another sample of individuals. Again, this has to be really as large as possible. And you count the number of individuals that are tagged. And then you use a bit of ratios. You use this formula to calculate what's called Lincoln's index. Basically the estimate for the size of the population, where you multiply the original number of individuals in your first sample Then you multiply that by the number in the second sample, and you divide that by the number of individuals that are marked. So it's kind of in chronological order if you use the formula. First sample, second sample, number tagged in the sample. There you go. Now, of course, there are quite a few assumptions that you have to make here. The first one is that you assume that the tags and the marks have not come off. You're assuming that this redistribution has happened. You're assuming that there has been no migration, there has been no increase in population size, things like that. So now let's move on to the second half of the topic of biodiversity, and this is the one that students tend to struggle with, mainly because it is a very wordy, waffly topic. And textbooks don't tend to do a good job on this either, but exam questions require you to be very specific and very concise, and you need to use very particular keywords. It really has to be nice and snappy and crisp if you're going to give exam answers, and that is hard. But I'll try my best in this video. So the next section looks at the factors that affect biodiversity. So the question is, what can cause biodiversity to reduce? And the obvious answer here is, of course, us humans. Human activity is a major contributor to the reduction of biodiversity. And specifically, that could be deforestation, agriculture, pollution, overexploitation of resources or populations, and the introduction of invasive species. So we're only going to look at deforestation and agriculture in some detail. Just remember that pollution can be both air and water pollution. Things like oil spills into the sea can greatly reduce biodiversity of aquatic habitats, fish populations. Overexploitation is when we take advantage, when we use nature's resources unsustainably. We'll look at sustainability in much more detail in later topics in the course, but this is basically just the using resources to meet our demands, but not the demands of future generations. And finally, invasive species are species that are non-native to an area, but are better adapted to this environment than the species already present. Which leads to the displacement of that original native species. And of course, any other species that depended on that native species will also be affected. Then we have natural disasters and climate change. So let's have a look at these three reasons, these three factors in a bit more detail. So the first one is deforestation. This is the major large-scale clearing or removal of forest or woodland by humans. So here we had a very nice, neat area of woodland, and then just one large chunk of it has been removed. Now, the trees that have been felled can be used as a source of timber. Timber is very important in the building industry. You can make furniture out of it, fences, houses, many things. Or the wood is simply burnt, it's used as a fuel. And then the land that has been left behind is used for building new roads, railways, airports, towns, villages, any kind of infrastructure, or it could be used for agriculture as well. So why does deforestation negatively impact biodiversity? So the first very straightforward reason is that if you're cutting down trees, you are removing individuals of a population, and you are directly reducing species biodiversity. That is quite obvious. The second problem is that you are destroying or fragmenting woodland habitats. So you are either completely removing a habitat, or you are breaking it apart into small pieces. And those small pieces can't link with each other. The isolated populations can't then interact. And so if you're forming small isolated populations, they could also reduce in their genetic biodiversity and could also decline in their size. And lastly, deforestation is a major contributor to climate change, either directly through the burning of wood that releases carbon dioxide because, well, as we know, trees store carbon. And likewise, because trees absorb carbon dioxide, they fix it as part of photosynthesis. They are reducing the concentration of carbon dioxide in the atmosphere. If you have fewer trees, then you have more carbon dioxide in the atmosphere. And there you go. So you are removing the store of carbon from the earth, really, and that of course contributes to the greenhouse effect and climate change. And climate change by itself also negatively impacts biodiversity. We'll look at that in just a moment. Now, although deforestation can be natural due to acid rain, for example, most of this is of course due to us, humans. Another human cause of low biodiversity is agriculture. And this is either farming or the growth of crops. So the problem is that the human population is continuing to increase, and it is doing so at an increasing rate. Likewise, the demand for food and other resources is also increasing. There are now more mouths that need to be fed, and this problem needs to be solved by agriculture. Agriculture needs to develop new techniques of increasing food production. And these techniques, of course, come at a cost to biodiversity. Here we have a very famous cartoon that illustrates the problem. You can see we have a graph that shows the growth of the human population, and the train represents agriculture, or the world food supplies. And you can clearly see that it is not coping with this increasing rate in population growth. So over the last century or two, agriculture has had to step in with these new strategies so that food production can be increased. As we've seen already, selective breeding is often used. That, as we know, reduces genetic biodiversity. But then another thing that reduces genetic biodiversity is a monoculture. A monoculture is when you have a complete field or plantain of genetically identical clones of a single crop, or at least the same species of a crop. And so there you're going to have a very small gene pool, and if they're all clones of each other, well then genetic biodiversity is almost non-existent. The other problem with agriculture is the removal of hedgerows. So previously fields were split by hedgerows, but nowadays with this increased demand for food they are in the way, and so they have been removed. They are in the way because of something that we call mechanization. This is when tractors and other machinery are doing the job instead of humans who go out and do the sowing and the collecting of the crops. And lastly, in agriculture, herbicides and pesticides are very often used. Herbicides are weed killers, and although weeds are plants that you do not want because they compete with your crops, they are still species, and so by removing them, you are decreasing species biodiversity, and likewise with pesticides. Fertilizers are nitrogen-containing compounds that are used to increase the fertility of the soil. Now, if it happens to be that these fertilizers run off into neighboring bodies of water, then that can lead to a process called eutrophication. Now, for OCR, you don't need to know much about eutrophication, but essentially this is when the nitrogen in the water is used by algae. Their population rapidly increases. They then use all the oxygen in the water. They form they rise to the surface of the river, and they prevent sunlight from reaching other seaweed, for example, in the water. And basically all the fish and aquatic organisms present suffocate and die. So yes, in any case, we know that agriculture can definitely decrease biodiversity. And now let's have a look at climate change. This is a very relevant topic to all scientists. Although there is a lot of overlap with geography, we should know about this as biologists as well. And hopefully nobody is denying this, almost nobody. Human activity is the major cause of climate change, and in particular that is the burning of fossil fuels. Climate change is reflected in the increasing global air and sea temperatures, the mean sea level, and then the fact that the weather patterns are now becoming more extreme. You might get very warm winters, cold summers, more rainfall or less rainfall. You might get hurricanes and storms more often, things like that. The weather is becoming more extreme. How does this impact biodiversity? Well, first of all, if we consider the habitats in polar regions, many animals like polar bears, for example, rely on these polar ice sheets, and they, of course, are going to melt if temperatures are increasing. The melting of ice causes sea levels to rise, and that means that any low-lying land near the sea can be easily flooded. That can also destroy terrestrial habitats. The other thing that you need to consider is that seawater is of course salty, and if that then flows upwards into rivers, it goes upstream, then freshwater habitats will also be lost. Now, if global temperatures are increasing, if periods of drought are going to be longer, then plants that are not adapted to these dry, arid environments will fail to survive. And so all the crops that we rely on now will also potentially become extinct. So with this expansion of dry, arid environments northwards and southwards, we will have to find new crops. We might have to use even more new techniques in agriculture to adapt to climate change. And because the weather is becoming more extreme, we have an increased frequency of natural disasters, and they by themselves can destroy habitats and cause population decline. Okay, so we've just seen how human activity can reduce biodiversity. But the question now really is, why should we care? Why bother? So what are the reasons for maintaining biodiversity? We can classify the reasons into three categories. The first one is aesthetic, then we have economical, and finally ecological reasons for maintaining biodiversity. The aesthetic reasons are the most simple and straightforward ones. These are closest to home and make most sense. Very simply, an area with high biodiversity will have more natural beauty. The landscapes will be more beautiful, tourists will be able to enjoy these landscapes, and of course tourism, as we will see in a moment, has economic benefits. But also you shouldn't forget that picturesque landscapes provide inspiration to artists, photographers, musicians and writers. And lastly, there have been many studies that have shown that being in a beautiful biodiverse environment can help relieve stress or recover from injury. So now for the economical reasons to maintain biodiversity. So we've just seen that tourism can benefit the local community and the local economy. You might have come across the term ecotourism as well, and this is becoming increasingly popular and important because the income that is generated from ecotourism is used as part of conservation of the ecosystem itself, and it really gets the local community involved. The locals work as tour guides, they provide accommodation and so on, and that gives them a source of income. Many locals might rely on deforestation or agriculture. As we've seen, they reduce biodiversity, and instead their income source is shifted towards ecotourism. So that is also very important. As we've seen with monoculture, that reduces, of course, genetic biodiversity. The reasons for maintaining that we'll see in a moment, but also that could result in this process called soil depletion. This is when you grow crops on the same soil over and over again, and all the nutrients in the soil are taken up by the plants, and fewer remain over time. And this is particularly amplified when you have the same crops being planted in this soil, because they will take up the same nutrients every time. And so just eventually the soil will be no longer, you won't be able to use the soil for growing crops. If you can't grow crops, then that's a decrease in food production, and that negatively impacts the economy, particularly if a country relies a lot on agriculture. Deforestation can lead to soil erosion, or this word that we can use here, desertification. Again, very similar to soil depletion, this means that land can no longer be used for agriculture. Now here's the important point. Many medicines, for example, have natural sources. They have come from tropical rainforests even. And so if we continue to decrease biodiversity, some of these economically significant species may become extinct before we even discover them. So it is important that if they become necessary in the future, we still leave them there so that they can be used should they need to be. And very straightforward as well, if you unsustainably remove or use resources provided by nature, then eventually that will lead to the collapse of whatever industry depends on those resources in that area. So if you continue to carry out deforestation, if you continue to fell trees, there will be no trees left behind. That will lead to a collapse in the industry of timber in the area. And lastly, one thing we shouldn't forget about is genetic material and how many plants and animals can act as sources of potentially important genes in the future. These genes can then be used as part of genetic engineering, maybe, to solve problems like increased periods of drought. So, one example here is that we can transfer a gene from a cactus or a similar species to our crops that we rely on now, so that then they become drought resistant. That is of course very simplified, but you get the idea. If we reduce biodiversity, if we continue with this same pattern, then we might lose this genetic material and we won't be able to carry out breeding like this in the future. Okay, and lastly now we'll have a look at the ecological reasons. This links nicely into what we said about the wall and removing of bricks on the wall. This is something that scientists call interdependence. This is when species in an ecosystem all rely on each other for their existence. And one particular type of species that are important are keystone species. These are ones that have a disproportionately large effect, importance in an ecosystem. And if they are removed, then they cause complete collapse of the ecosystem. And one example of keystone species are the grey wolves that were reintroduced into the Yellowstone National Park in the States in the 90s, I believe. And previously they were absent, and after their reintroduction, everything went back to normal, basically. Okay, so we've answered two questions: how biodiversity can be reduced, why we should be bothered about that. So how do we achieve this maintenance of biodiversity? What are the methods that we can use to maintain it? And the main method that we look at is conservation. Conservation is a key word for A-level biology, and it means that humans are actively involved in managing ecosystems and protecting them so that endangered species don't continue to decline in their population size. The main idea of conservation is to reduce the effect of any biotic or abiotic factors on a population that will limit the growth of that population size or even cause it to decline. So we're making conditions as favourable as possible for an endangered species population. Conservation in situ takes place within the natural habitat of the species, whilst ex-situ conservation takes place outside of it. So let's have a look at a few in-situ conservation techniques. The first one is the reintroduction of native species. So maybe after a captive breeding program, the bred population returns to its natural habitat, and then the original population size can be resumed. Also, the removal of invasive species, the removal of competitors, which is a biotic factor affecting population size, can also have positive impacts on the endangered species population size. Then we have this idea of controlling or reducing overexploitation in the form of hunting or poaching. And that does prove to be very difficult. It is very hard to enforce the legislation and police areas, police the habitat and ensure that no poaching takes place whatsoever, but it is still nevertheless one important technique. Then human access can also be restricted. This might be in the form of seasonal bans where you are not allowed to enter a national park maybe at a certain time of the year when there is a breeding season for this endangered species or something like that. And then simply you can feed the endangered population, you provide food resources, and that reduces intra-specific competition when the individuals of the population compete with each other for food resources. Controlling grazing is also important, and again another key term that you will come across later in the course, succession. So for now, just probably it's better to memorize this method of in situ conservation. Now in relation to ex situ conservation techniques, the first one is botanical gardens. This is when wild plant populations can be kept and managed in these gardens, where they are cared for and treated, ensured that they are well watered, that they are free from disease as much as possible, and again when the time comes around they might be able to be reintroduced into their original natural habitat. Then a similar technique is a seed bank, and this is when seeds from all over the world are kept in a controlled, in terms of temperature, environment in this container. This image shows the seed bank in Norway, in Svalbard, and it is important that the temperature is kept low so that the seeds don't germinate. And so in the future, you never know, the genetic material of a seed might become important, as we said already, maybe in genetic engineering. And lastly, as we've seen already, captive breeding programs. So this is when an endangered population is transferred to maybe a zoo, and they are cared for there, and the population is bred. Now there are many problems with captive breeding programs, and the first question that you need to think about, well, what are you going to do with this captively bred population? You're probably going to return it to its original natural habitat. But it could be that the habitat itself was the very cause of the decline of the endangered species population, and so you would have to restore or reclaim the original habitat before reintroducing it. The other problem is this idea of domestication. Any offspring of this captively bred population will be growing in a controlled, in not a wild environment, and so they won't learn by themselves how to hunt, how to survive, and so when they do get reintroduced they will struggle. And lastly, now we need to think about some international agreements and treaties that are made to maintain biodiversity. It is quite obvious that animals and plants don't respect countries' borders, and so countries need to work together to maintain biodiversity. So first of all, we have the International Union for the Conservation of Nature. This is essentially one of the largest and most important global authorities that oversees all the international treaties and regulations for maintaining biodiversity. It consists of many governmental and private organizations, scientists and experts that constantly research biodiversity and publish things like the Red List of endangered species and so on, and tries to work out ways of maintaining biodiversity through the techniques that we've just looked at. The next important global agreement was the Earth Summit that took place in 1992 in Rio de Janeiro in Brazil. And the results of that summit, of that meeting, were called the Rio Conventions, and they looked at biological diversity, climate change and desertification collectively. And this is when 172 countries came together in Brazil to establish these new protocols. One other important international convention is CITES, which is the Convention for International Trade in Endangered Species. So this looks at forming and enforcing laws to do with trading of species that are endangered. Some categories of animals and plants cannot be traded at all. It is illegal, while some are allowed to be, but with restrictions. And lastly, it is important to realise that biodiversity can be maintained at local and national levels as well. And so the example that you need to know is the Countryside Stewardship Scheme that was introduced by the UK government in the 90s, I believe. And this just offers financial incentives to landowners to basically look after the environment. Farmers and woodland owners can apply to get this countryside stewardship, and then they use these named techniques, methods of maintaining biodiversity, in exchange for this funding from the government. Okay, so that is it for the topic of biodiversity. I appreciate that a lot of this topic, particularly the second half, is quite abstract and waffly. So I actually recommend that you watch documentaries, maybe you read more about these international agreements and methods of maintaining biodiversity, rather than just reading the textbook. You can do some research online, do some wider reading, and that will help with your understanding. And it will make this a bit more, you'll be able to visualize this a bit better. If you have any questions at all or any feedback, please feel free to leave them in the comments. Thank you very much for your attention. Good luck and goodbye.